Folks, hopefully you can hear me. My name is Stan A. Hall. I'm a professor here in computer science and also the director of forensic science. Great pleasure to introduce David Shaw for the Triangle Computer Science Distinguished Lecture Series. David is one of the most well-known names in computer science, and that's for very good reason. He pioneered the use of high-performance computing in the financial markets, and he's now having a similar transformative impact on HPC and its uses in molecular dynamic simulations. David earned his PhD in computer science in 1980, I believe, from Stanford, and served on the faculty of the Columbia University Computer Science Department until 1986. He founded the D.E. Shaw Group, which is a big building on Wall Street, in 1988, and since 2001, he's also had time to run a hands-on computational biochemistry research program at D.E. Shaw Research. He was appointed to the President's Council of Advisors on Science and Technology, that's PCAT, by President Clinton in 1994, and was reappointed again by President Obama in 2009. He was elected to the National Academy of Engineering in 2012. He is a fellow of the American Academy of Arts and Sciences and a winner of the prestigious Gordon Bell Prize. His recent work focuses on one of the great challenges of our time, and it spans computer science and biochemistry, how to accurately simulate the movement of protein molecules for more than just a few microseconds. Dr. Shaw and his research team developed Anton, which is a special purpose HPC machine to address this problem. The D.E. Shaw research team work led to Science magazine naming the machine and the research it enables as one of the 10 most important scientific breakthroughs of 2010. On a personal note, I want to tell you that David is one of the most gentlemanly, and pleasant people to work with you'll ever meet. And it is truly a great joy for me to welcome him to this distinguished lecture series.
Thank you, Stan. Thank you. The last part means a lot to me. Let me just start by telling you a little bit about what we're doing at D.E. Shaw Research. As Stan says, I abandoned Wall Street about 11 years ago to spend full-time on a problem or a set of problems that was intriguing to me as a computer scientist, even though I really had no background at that point in biology or chemistry or any of the areas that turned out to be necessary. And that was the question of how we can simulate the motion of biologically important macromolecules, just big molecules in the body that are important in various ways, and in particular protein molecules, which are key to almost everything in the body. Those and nucleic acids account for a lot of what runs our bodies, both in terms of proteins as structural units and also as signaling units, things where you'll have one protein that attaches to the other and causes it to change shape, which in turn means it can now signal to another one. And you have this big sort of complex wiring diagram of signals that are transmitted. And then there are things that have holes that go into the membranes around cells and let some molecules through and some others don't get through. Those are proteins also. There where a lot of interesting biology happens. And the way I sort of fell into this was that a good friend of mine, Rich Friesner, who's a chemistry professor up at Columbia, started telling me about the research he was doing, and our daughters have always been and are still best friends. And neither of us is very good at vacations, so when we got together with the family, we'd take long walks, and he'd tell me about, this is the hard part, we can see these simulations, but not for long enough to really see what's going on with the behavior of these proteins. So having, when you have a hammer, everything looks like a nail. I had remarkably few hammers at that point. What I had done at Columbia and in my doctoral work also was to design and build massively parallel machines. So the first thing that occurred to me was, can you build a special purpose architecture that was designed in a very different way from an ordinary computer that could vastly speed up this problems to get me to the time scales that Rich said were really important. And that's what D.E. Shaw Research focuses on. There are a couple little related problems, but basically we're interested in the use of what are called molecular dynamic simulations, which are basically just following how a protein moves over a period of time, and then using that to discover how interesting things about how proteins move, how they interact with other molecules, and then the ultimate goal is to try and understand biological mechanisms, translating the structures and those motions into a better understanding of the underpinnings both of normal physiology and also disease. When something goes wrong, when there's a mutation in a protein, when it encounters something else that gums up the works, what's going on? And more importantly, and this is the long-term focus of D.E. Shaw research, we'd really like to cure some diseases. Have some way of understanding mechanisms and then designing molecules, and in particular, typically small organic molecules of the sort that most drugs are, designing ones that can intervene in diseases and cure people or extend lives or improve the quality of life. So we use our hammer, which is molecular dynamic simulations, to study these problems and try and come up with something that can actually be useful at some point. So let me just take you through first some of the underpinnings. I just gave you a sort of quick sketch of proteins, but I want to explain where they come from and then how they are structured and how they move. So let me just give you a little bit of a refresher course in basic biochemistry. So for a start, DNA codes for proteins. We now know it does a lot of other stuff also, but it's fairly simple code where every 3 base pairs together codes for one of 20 amino acids. It could code for more, but some of it is redundant. And there are 20 of these types of amino acids. They're linked together just like beads on a chain. So it's literally that simple. There are 20 colors. The beads go in a certain order. And once that happens, that determines the 3-dimensional structure that they have in the body. And there's sort of different levels here, and I'm mentioning this largely so that you can interpret some of the pictures and movies. You start with this structure. It folds up typically into, most of it, into some intermediate structures, which can be helices or sheets that lie next to, strands that lie next to each other and form sort of a flat piece of paper-like sheet. And then the final stage is those things typically fold up into a sort of globular shape. They take on some characteristic shape, and we know experimentally that for most proteins, even if you do this in the test tube, if you put in this string of amino acids, it'll fold up into that correct shape. It's not true for quite everything, but to a first approximation, that's what you can think of. So it's DNA to beads on a chain, folds up into a 3-dimensional structure, and that structure moves and does interesting things. So where are we? Well, we've decoded the genome, and even that's sort of an oversimplification, but that was a huge breakthrough scientifically, and it's driven a lot of advances that we have now. But what that basically gives us is something like a parts list. We have these identifying numbers. You can sort of look them up, but for a large fraction of all of those genes, we don't know what the proteins are that come out of them. And even when we do, we often don't know the structure. For many proteins we do, we're getting more all the time, but some of the most important ones, some of the ones that live in membranes and control active switches and control a lot of different things, we really don't know what they look like, much less how they all work together, and that's what you'd really like to know. So one of the problems is the question of determining structure, but also the question of how does the machine actually work? So how can you do that? Now this is sort of a computer scientist's interpretation of the world, which is there is computer science and then there's everything else, which is all mysterious. So, you know, from my viewpoint, laboratory experiments, which were the traditional way of getting information about most things biological, the basic principle by which they work is magic. And what you do is you come up with fancy instruments. Many of the things are wet. You shoot high energy particles at things. You watch them vibrate and a lot of things like that. And magically you get data out of there. This is obviously impossible. So that was ruled out as a possible paradigm for research in our lab. And in addition to that, it's very hard to get certain sorts of information. Experiments are really, really powerful and can get a lot of information that we can't get in our lab. But at the same time, it's very hard to get detailed moving pictures. It's hard enough to get still pictures of some of these structures. And the amount of information we have about exactly what the motions are that proteins go through on a biological timescale is very, very primitive. Whereas simulation, on the other hand, if we start with a structure, if we try and fill in the gaps and see, well, it moves from this known thing to this known thing, if we try and see, when this happens, this other thing moves, that's the kind of thing where if you have a simulation and all of the data is recorded there, you can take a look at it, rerun the movie, go backwards, study the part that you want. That's a very appealing way to get complementary information. And in some cases, we think of that as a hypothesis to be validated by experimental tests or refuted, as the case may be. So how do you do those kinds of simulations? You can very accurately simulate tiny molecules, but for molecules the size of a whole protein moving in a solvent environment, something that's mostly water with some ions floating around, you have to use some approximations. In fact, you can't solve the Schrodinger wave equation and get exact solutions for pretty much anything. And in this case, you have to use some pretty severe approximations. The state of the art in that area is what's called molecular dynamic simulations with what's called a molecular mechanics force field. It's a classical model of the underlying physics. And I'm going to show you what one of those looks like now, not because I hope that anybody's going to go home remembering any of the formulas, but I'm trying to do this mostly from a computational viewpoint. Actually, incidentally, can I just ask quickly, are most of you computer scientists? How many are computer scientists and engineers and so forth? Okay, and then biologists and chemists and other magicians? Actually, more evenly balanced than I'd realized. Okay, good. I'll try and shift a little bit the balance of who I bore for how much of the time. But let me just run through a quick overview from a computer scientist viewpoint of what the algorithm looks like. And it's actually fairly straightforward. It's just very computationally demanding. So when you do a molecular dynamic simulations, this is what you're doing. You start by dividing time up into a lot of tiny time steps, and then you're going to integrate Newton's laws of motion over this time by, in an iterative way. The problem is these time steps have to be very small in order to keep the equation stable and not come up with crazy numbers that where the energies blow up and all the atoms go flying off in random directions. And the time scale that you have to simulate at with straightforward techniques are on the order of a femtosecond, which is 10 to the minus 15th seconds. So whatever you have to do a lot to simulate any reasonable length of time. And what you have to do in each one of those time steps is conceptually, it's not quite as bad as this, but conceptually, for every pair of atoms in the system, you're going to calculate the forces exerted by all of the atoms on, let's say, this atom. So it's a quadratic problem. You're looking for all possible pairwise interactions. And the way that, the forces that you look at are ones that are modeled by this molecular mechanics force field I mentioned. This is what one looks like, and I'm not going to go through most of it, but in every step, we're going to evaluate something that looks very close to these equations. There's several different force fields, but they usually have almost exactly this kind of structure with minor variations. So the first three are what are called bonded terms, and those express the forces, they approximate the forces between covalently bound atoms. The first one is two that are connected directly to each other. Second one is if you have three atoms, the angle between them. And then the third one is a torsion angle. So if I had four atoms, the way they twist around. And there's some preferred positions, preferred distances, preferred angles. And there's sort of a harmonic penalty here and a trigonometric one there for being too far away from it. So this sort of pulls things that are bonded together back to preferred states and penalizes them if they're too far apart. That's not the major problem in doing MD simulations. The major problem is with these non-bonded interactions, which are interactions between pairs of atoms that are more than, I guess, 1, 2, 3 or so apart, 3 atoms apart. but which can be near each other in space and exert strong effects on each other. So the simplest one, which I'll bet most of you will remember, is Coulomb's law. This is what I'm showing here is actually not forces, it's the potential. And so it's the gradient of this that determines the forces, but the potential energy between them is proportional to the two charges. If it's two positives, they want to repel. If it's positive and negative, they want to go together. Multiply those together and divide by the distance between them. And this isn't a conceptually complicated part of the formula, but this is what actually takes most of the time in an MD simulation. It's just the electrostatic interaction, Coulomb's law. And the problem is that the 1 over r falls off pretty slowly. It gets smaller, but you can't ignore the interaction between a positive and the attractive force between a positive and a negatively charged atom within the length of, say, a protein, or even part of the way out into the water. So this takes a lot. And again, you're doing this quadratic number of, it's quadratic in the number of atoms, and you have a lot of these time steps. And then to a lesser extent, this term of what's called the van der Waals interaction, this part basically, the 1 over r to the 6th term basically just means that independent of charge, atoms sort of like to be closer together. And this part here says, but not too close, the 1 over r to the 12th part. This is not the correct functional form. It's a rule of thumb that people have sort of fitted to. In fact, this one right here is a quantum mechanical weirdness, which is based on the idea that electrons are fermions, and so their position in this 3n-dimensional space is such that if they get very close to if the electrons, basically you have an anti-symmetry property where if you interchange two electrons, the wave function has to be inverted, and so if they're right on top of each other, it has to go to 0, and there's sort of a smoothness thing that's imposed. It's weird quantum mechanical stuff, but the way to think about it is you really can't let any two electrons get too close, which means you really can't let two atoms get too close to each other. And it doesn't look anything like this, but that's true of many of these things. They're gross approximations that happen to be fairly close enough to get surprisingly good results in at least many cases. And my very last slide is going to be, but not all cases, and we don't know just how bad it really is. Anyway, we'd gotten, just to refresh your memory, we divided time into tiny steps, calculate all these pairwise forces, then for each one of those pairs, evaluate something that looks like this, and then that gives us forces. We move each atom a tiny bit, and then we repeat the process over and over again. And the problem is that over and over again is a lot of times. So the target that I was interested in when I first organized the group was to get to timescales like a millisecond or so. And at the time we started it, that was about 3 orders of magnitude longer than anybody had been able to simulate. Now it's about 2 orders of magnitude longer than had been done previously. And the reason for that was that that's a time frame at which a lot of interesting things start to happen biologically, and that just hadn't been accessible in some cases even to experimental work, and never before to simulation. So why do this at all? Well, let's just set up some things that are quite extreme, not true. What if it turned out that the force field that I just showed you was perfectly accurate? It just solved the Schrodinger equation. And then what if MD simulations were infinitely fast, so you could simulate for whatever period of time you wanted? Why would that be interesting? Well, one thing would be, if you wanted to know the structure of proteins whose structures aren't known, you would just sort of throw them in there just like you did in an experiment, watch them fold up into the structures that they have, watch them interact with each other, watch how they move, and you could stay out of a laboratory with all of this mythical equipment and simply do data mining on all this perfect data that showed how all of your molecules was going to move. Now, the problem with that is that this isn't true. Like, MD is definitely not infinitely fast, and it's also not the case that these force fields are perfectly accurate. But we've now seen, in fact, that when you make this, the length of time you can simulate significantly longer than it was before, we can actually model some sorts of things that had been inaccessible before. And those primitive force fields actually are good enough to tell you some interesting things. So what happens in that time frame? Well, one thing is that big structural changes often occur at times far longer than what was possible before this machine that we built. The record length simulation before was 10 microseconds, which was done by Klaus Schulten on one of the world's most powerful supercomputers. And there was a huge amount that was learned by simulations that length and shorter, but a lot of the kinds of things we were interested in were on the order of between that level and 2 orders of magnitude longer. And over that time frame, we know through a lot of experimental data that major structural changes in the protein often happen. You know, it'll go from this shape to something entirely different. And you just don't see that if you're simulating a very small amount of time. Also, a lot of interactions with proteins, between proteins and other proteins, between proteins and nucleic acids, DNA and RNA, and importantly to us, interactions between proteins and drug molecules. These things very often, in the changes in shape and in dynamics that they induce, those kinds of things often occur over time frames that are significantly longer than the previous 10 microsecond maximum time scale. And the final one is probably the most famous of the problems throughout this whole area, which is called the protein folding problem. And protein folding is actually, it means different things to different people, and the part that I think most people hear about and associate with that most is just predicting the three-dimensional structure of a protein from its amino acid sequence. But the part that scientifically is probably of more interest to people in the field is how the process of folding works. Now, folding is a process by which this long chain of amino acids folds up into a three-dimensional structure. And understanding the process, what drives that? Does it go, does it always go the same way? Does this happen first and then this and this all the time? Or does it sometimes go here? Does it stop and get stuck and then back up? Very little was known about that. And so that was one of the things that we wanted to simulate. Folding also sometimes does go wrong in various situations, and misfolding is implicated in various diseases, but it's probably best known both as a scientific problem and because it's a very useful task, a very useful hard task that can help us with a lot of the other changes that are important scientifically, but also in terms of potential therapeutics. So what to do about this 2 order of magnitude shortfall? Well, our hammer was something called Anton, Anton 1, and this is one of the machines. This is a 512 node machine. These are based on custom ASICs that we designed, and so this is 512 of those ASICs and the surrounding stuff around them. And what Anton is a special purpose supercomputer, if you will. It just is for molecular dynamics simulation. It's not a general purpose supercomputer. If we knew how to design a machine that could do any kind of scientific simulation, orders of magnitude faster, it would be a great service to the world, and we would certainly try and tell everybody about it, but I have no idea about that. We're able to have the luxury of wanting to do just one thing, but much, much faster. And Anton is, in fact, about 100 times faster, as I mentioned, than was previously possible with a general purpose machine, but it's very brittle. It's not, in theory, you could program each of the nodes, and they've got some general purpose processors on them, but that wouldn't be a smart thing to do, wouldn't be very efficient, and it would be really difficult to program because that's not what it's designed for. But Anton was designed for just this one application, and together with a very different kind of algorithmic approach on several levels. And I'll say something about that a little later, but I want to first give you a shot of just a few movies showing you what an MD simulation really is and what you might use it for. So Anton, it's been in operation for gee, I don't know, 2.5 years or something, I think. I might be off a little bit. We built 13 machines, of which, let's see, all of them are the size you just saw, except one is twice that size and one is 4 times that size. And each one of them is capable of millisecond scale simulations, which we run on a regular basis. Most of the runs that we do are shorter, somewhat shorter, but still long by conventional standards. But sometimes when we need to, we'll take a look at how a molecule moves for a very long period of time. So for those of you who are involved in this area, an obvious question is, well, a millisecond of what? A millisecond simulating what system? And there are a couple standard benchmarks, but one of them is dihydrofolate reductase, which is a system that has about 25,000 atoms, including water molecules. What we do is we simulate, sometimes you can use an approximation of the water environment without representing every water molecule. We do, because we happen to be interested in some interactions of water molecules with the surface of a protein, but you can learn a lot of interesting things without that as well. So those are the ground rules. And then the other part, which is very important from a computer science viewpoint, is what we're interested in is running very long single simulations. Now that's a much harder problem, unfortunately, than running a very large number of short simulations. And that's not to say that that isn't an interesting thing to do. For example, Vijay Pandey at Stanford has done some very interesting work where this is a great model. What he has done is gotten people interested in the problem of protein folding, and they've not only dedicated their computers and now their video game machines to running with their spare cycles over the internet, but there's a reasonable number of people at this point who are actually buying new Xboxes or whatever they are now, my kids could tell me, connecting them to the internet, and they buy them just so that they can contribute to this project. And as a result, Vijay has managed to mobilize huge amount of computer power for something that's extremely worthwhile. But what he's got is a bunch of short simulations. 200,000 of them, I think, on average, are connected. The question is, how can you combine all of that and get something meaningful? And each time I've said, well, it's very interesting you get this, but of course you couldn't infer this. Vijay has found a way to do that, and so I've stopped. I'm now out of the business of saying things that Vijay can't do with his distributed processing approach. But I think he would be the first to say that that's a different kind of set of things that you can do. There's some purposes for which you really have to be able to see one long trajectory rather than combine information from a lot of short ones to answer many important biological questions. Now, why is it hard to, I should mention the other way that you could make the problem easier is if you were simulating very large molecules and you wanted to get the same speed up on a per atom basis, that's a much easier problem also. And in both cases, the reason is that the thing that becomes a bottleneck pretty quickly, as in most parallel computation, with the exception of what are called embarrassingly parallel problems, but the thing that hits you as a bottleneck is interprocessor communication. How do you communicate between all these different processors so that you can break the problem up into a lot of pieces, cooperate to solve one big problem together? And very often what happens is you have more computational power available on the chips, especially as things move. The way Moore's law is turning out right now is that we get more and more silicon real estate. We can have more stuff going on simultaneously on a chip, but the speed with which we can communicate between chips is not growing anywhere near as fast. So this is what's, in various forms, this keeps rearing its head when you try and accelerate a process by having, a million cooks in the kitchen and trying to get your eggs cooked in 4 seconds. It just doesn't work at a certain point. So interprocessor communication, not surprisingly, is a lot of what we focused on. And ultimately, if I don't run out of time, and I think I'm okay, I'll say something about some of the things that we've done to minimize that and the way you have to alter the computation to allow you to do that. let me mention one other thing that I always forget to mention. One of the things that we want to be able to do is to observe long molecular dynamics trajectories, see proteins move for long periods of time. But there's another thing that I didn't really anticipate the impact of until, we saw how group members were actually using it. And that is for rapid turnaround. It turns out that, sometimes you just say, I want to run this simulation. I have to run it for 500 microseconds of, simulated time. But a lot of the time what you do is you kind of play and you say, let's simulate this, just, see qualitatively what's going on here. And then you see some interesting motion. You're saying, I wonder if that's real. Is that repeated? Okay, run one long simulation. And typically it's even worse than that. You try one thing, this fails, you look at that, you play it back, you run this, you try other runs. If that process involves even if you had like one desktop computer and it was very cost effective, if it takes 3 months every time you do that casual experiment, then in real world time, you're never going to get to the important experiment. So part of what turned out to be important was simply being able to run what was previously a very long simulation, over a matter of an hour or so and get the feedback and decide what's the next thing you want to do based on that. So this is just a quick summary of the absolute lengths. This is Schulten's result on what's called the WW domain. And we actually, when Anton 1 was up and running, one of the first things that we did was try this again and do a simulation of the WW domain. And actually what Klaus was looking at is the protein folding problem to see if he could get the amino acid sequence for this particular protein domain, a chunk of a protein, to fold up into its native structure, the one it has in the body. And it didn't fold. And he was able to show that the reason was not only because he hadn't simulated long enough, he had good evidence that there were force field defects that were involved also. So for various reasons, we wanted to test that. And so we ran a simulation that was over a millisecond long, and we're able to get the WW domain to fold and unfold repeatedly. We actually had to improve the force fields using some quantum mechanical data that we'd analyzed over a period of a couple of years in our lab. But when we did, we were able to see reversible folding and unfolding. I'll just explain quickly because there'll be a couple references to this later. A protein will, left to its own devices at biological temperature, it'll fold up into this three-dimensional shape. But if you heat it up, at a certain point, it'll sort of fall apart. It will denature. And there is something called the melting temperature, which is the temperature, roughly speaking, where it'll be folded about half of the time. So what we did is simulate it at that melting temperature, which is a temperature at which we would expect to see it fold and unfold and fold and unfold enough times that we could get statistically significant results about the pathway of folding. What happens first? How does it work? What moves at a microscopic level? Those kinds of questions. And with these changes in the force field, we were able to observe that. And then the current longest simulation, I think at this point, is still about 2.2 milliseconds, although we have one running now that may be longer at this point. I'm not sure. Just one other quick thing for those of you who are involved in, actually could be computer scientists or interested in biology or biologists who do computational work. That's that one of the Anton machines we built, we've made available free of cost for nonprofit institutions for research use. And we gave that to the National Resource for Biomedical Supercomputing, which is at the University of Pittsburgh, and there's a connection with Carnegie Mellon, which is of course near there. So far, this was just changed, but there have been 70 research groups that have made use of it, and the National Academy of Sciences allocates the time and gives it away to people who want to use it. And one of the things we're interested in is to get people involved in doing simulation who haven't been doing that before. Among other things, they can think of a lot of stuff that just wouldn't have occurred to us, and many of them have expertise with molecular systems of a sort that we don't. And from our viewpoint, we sort of feel like, because we'd like to design drugs at some point, anything that helps the community of molecular dynamics simulations is great for us, you know, and hopefully it'll turn into a tool where we'll understand better than we would have by ourselves what it can do. So if anybody is interested in doing this kind of simulation, the second-year competition just happened, and they announced the winners a few months ago, I guess, but we hope to be able to continue this program. Okay, let me show you what MD simulations actually look like, and then I'll talk a little bit about what makes Anton fast, what the strategies were. So first, I'm just going to show you a simulation of a molecule. It happens to be something called GPW, which was not particularly of interest to us biologically. In fact, this simulation, I think, was run primarily to test out the machine. There were a lot of statistics and things about stability and other things to ensure that it was not, that we weren't seeing accumulating errors. So it's one of the first that we ran. And what I'm going to show you is, first of all, it's an idealized model. Each one of these little balls is an atom, and they're color-coded for the atom type. What you're going to see is sort of a smoothed version, because this is such a long simulation that if we were actually showing you all the motion of the individual atoms, you'd just see sort of a blur, because there's a lot of high-frequency vibration and so forth. But I'm going to try and give you a sense for how long a period of time we're simulating compared to what was going on before. So this is what an MD simulation looks like. And you'll see little things happening, little rotations, but then every once in a while it makes a big move. And those kind of moves were never seen in previous simulations because they just weren't long enough. Sometimes it'll just extrude out a lot and then tends to pop back into its original folded native state. And it's funny because, you know, we sort of knew what to expect by this point, but the first time I saw, it was actually not this animation, but a related one. The first time I saw one, it was kind of like, oh, that's what proteins are doing. Because no matter how much you read about it, just watching a movie, you get a slightly different feeling for what it's all about. Now, let me just, by way of comparison, show you what Schulten's previous longest simulation, the 10 microsecond simulation, would look like on this time scale. I'm doing this by hand, but roughly this. It would be about that. So there was a lot of information. And again, if we weren't sort of low-pass filtering it, you would see a lot of vibration, but you would completely miss the big structural changes that we're particularly interested in. Let me just show you one. I talked about the WW folding. This is one of the folding events. You know, it goes back and forth. Here's just one of them so you can see it. In terms of the representation, before I was showing you the individual balls, and here, this is what's called a ribbon representation, where you're just seeing the polypeptide chain, the string of beads moving around. And when it's curled like that, that's a helical structure, and then sometimes it's random. And what you're seeing here in gray is the native three-dimensional structure. That's what it actually looks like in your body. And what we do is start it here in just a random extended configuration, and you'll see it fold. And this one, I think I should really smooth this more because it's a little jerky, but you can see it trying to find the right position, again, based on the laws of physics as expressed in that force field. And sometimes it gets close, pops out, it won't be quite right. When it gets in something that's very close to the folded state, it's mutually stabilizing. There are a lot of interactions that sort of are mutually reinforcing. And you'll still see some wiggling around of the tails, and that's something that apparently really does exist. It correlates with a lot of experimental data. But basically, it's going to settle into its three-dimensional structure. And I think I eliminated the slides, that would show that. But the kinds of things that we were able to discover was the order of formation of some of these helical structures and whether it followed a single pathway. And the bottom line is, yes, it appears to for the most part. And other sorts of technical things that have been debated for many years. We don't have a comprehensive answer here, and this is a good time to switch to the next slide. The one before was a year before. This is another science paper that was just last year, where we were able to do the same thing for a systematically chosen set of 12 proteins. Bill Eaton had categorized a lot of the fastest folding proteins, and we took one representative one from each of a set of different structural categories, and we added actually 2 because we wanted to make sure our force field didn't prefer some things over others. So we added some mixtures of different kinds of structures. And what you're seeing here is, I can probably figure out which is real and which isn't if I spend time. Fortunately, it's not easy to do that. But one of these is the native structure. That's embarrassing. I can't remember which. And the other one, probably the red, is the structure that it folded into when we did a folding simulation. So for a very diverse set of proteins covering not all proteins, and it's a very systematic and perhaps biased set, which is simple, small proteins that fold very fast. And maybe there's something systematically different about the way that class of proteins fold. But that caveat notwithstanding, we saw all of them fold correctly. One of them is known to be not thermostable, and you have to, with certain mutations, it's more stable. The one that wasn't, in fact, we did find it had problems finding the right one, but the variant that's known experimentally to fold reliably does here too. So this told us some important things about most importantly, the quality of the force fields, the quality of the physical models, not just that we use, because we've just made minor tinkering based on evolved physical models that have come out of the community for a period of decades now, but that those force fields that the whole field tends to use actually are sufficient to reliably fold proteins with different kinds of structures. These are the helical structures, and this is actually a mixed one. I guess a simple one with beta sheets is this one, and successfully get them to fold. Now, big question is, how is this going to apply to more complex proteins? So far what I've said is we can't infer from this that it's going to work for complex proteins also. Personally, I'm a little more pessimistic than that. Although we haven't published all of this yet, we're gathering some data now in our most recent studies that suggests, although it doesn't show conclusively, that for more complex proteins, probably even if we could simulate long enough to match their experimentally observed folding times, the machine wouldn't get the correct answer, and that would be a force field related problem. And it takes a little time, but afterward, if anyone wants to talk about it, I can tell you why I very strongly suspect that'll be the case. And what that means is that more work is going to have to be done on force fields within our own group or other groups. And it also means that the future versions of Anton have to be able to handle some of the kinds of changes that we think are likely to be necessary. Probably Anton 1 would do it fine. We would just have to figure out what has to be changed about these physical models, but there's no guarantee. And it could be that we all find, hey, these parts of that force field that I showed you before are just fine, but here's this part that we knew was an approximation, and sure enough, nature really doesn't like it. This is not modeling it. Nature has the nightmare case is some funny quantum mechanical thing that it uses in making natural proteins, and that if you don't get it much more right than it is now, you'll never model the free energy landscape of the proteins, how it finds the preferred, energetically preferred structure to be in, and hence the changes that it's going to make. We don't know. I don't even have a hunch about how far we can push it, but we know that there are limitations now, and that maybe that there are going to continue to be limitations, that there'll always be limitations, but ones that are biologically significant and could give us a distorted idea or none at all about certain biologically relevant phenomena. So that's where we are at this point. And let me just show you a couple more examples and then talk a little more about the computer science-y stuff. I'm not sure exactly why I threw it in. It's just kind of a cool picture, but this is a potassium channel. It's a protein that passes selectively potassium ions through a membrane from the outside, through a membrane that surrounds a cell, or it can happen in other kinds of internal membranes as well. And so what we were able to do for the first time is actually to see the process of conduction of ions through there and see how it works and where the things sit And I'm actually showing you just the port, what's called the pore domain, the part that's the tube, but there's a little thing here that detects the voltage in the surrounding environment, causes us to open or close. We're going to have a paper coming out very shortly that that shows for the first time what the closed state looks like for this ion channel. We think it's a pretty general observation, what the sequence is, how the thing closes, and so forth. But this one is just the ions going through. And we also, looked at things like the rate at which these things pass through. You'll see there's some little water molecules in between the ions, and we wanted to know what the preferred pattern is. Usually they're interleaved, but not always, and a variety of other questions. These little carbonyl groups move around and sort of hold it in various positions and other stuff, but it's just fun to watch that kind of movie. And then here's just a couple quick things more on the applied side. One of the classes of compounds that we studied are proteins called kinases, which are responsible for phosphorylating other molecules, which can then act as a signal to drive all sorts of other things. Changes in other proteins and things going together and apart. And in particular, it turns out to be that this class of proteins is very central in many forms of malignancies. So this particular one, Abl kinase, is known to be involved in chronic myelogenous leukemia. And in fact, there's a classical mutation called the Philadelphia chromosomal mutation. And what you have in this kinase is a part that switches into an active state that causes the uncontrolled proliferation of cells, and patients die, and then a control region that takes signals in and keeps it mostly turned off, but turns it on when it wants that replication to occur. In this case, it gets lopped off and fused in a way that the regulatory part is missing, and it's just always turned on. So there's a drug, we didn't come up with this, but something called Gleevec that's imatinib, that is one of the exciting success stories in the field of cancer, of which there are precious few, unfortunately, where patients who used to die of this disease, many of them are living for years now, and in fact, we still don't know how long they'll survive. But this drug does something, and it was known that this switch involved that when the protein, the kinase, turned on or off, there was a characteristic 3 amino acid group here called the DFG group that moved from the in to the out position. I'll just show you quickly. You're seeing a flip occur, although it's a little hard without seeing this in binocular vision to see what's going on. But that flip causes it to move between a cancer-causing and non-cancer-causing state. And so what we were interested in doing is figuring out how does Gleevec work? What is it doing? Turns out it blocks this transition, holds it in an inactive state in a very particular way. And there are some other residues whose protonation state controls that flip. And based on building the structural model and this mechanistic model for this malignancy, we were able to make a bunch of predictions for experiments to do that we couldn't do, but we collaborated with John Currian, who's an experimental expert on kinases, and we predicted that if our model was right, then if you change the surrounding pH, then the binding of Gleevec would be enhanced, and if it's the other way, it would be inhibited. We also designed some mutations and said, if you made this mutation, we predict that it'll change in the following way. John then ran those experiments in his lab, and it matched very well. This is a kind of a model for our collaborations. They're almost always with experimentalists. I wish we could do some of those things in-house, but we don't have any experimental capabilities of our own. And the nice thing is it's not just we come up with hypotheses and then ask somebody to validate it, but the experimentalists we collaborate with almost invariably know more than we do about the particular biological system that they've studied for, often their whole academic careers, whereas we, do simulations on various types of molecules. So a lot of the time, they'll look at something and we'll say, what do you think about this? We observe this, and they'll say, that doesn't mean anything. But wait a second, can you rerun that? John did this once, and he said, replay that. replay it again. Can you just slow this down? That looks like it's about 3.5 angstroms. Is there a salt bridge forming there? And he knew right away what to look like. To me, it was a lot of balls and things and graphs of how far apart things were. He said, what would happen if you do this? So we tried experiments, and wonderful interactive process. And that's the kind of collaboration we really enjoy. There are a few labs where they do both, including some people here, and that's a really powerful combination. Anyway, so that's kinases. Last thing I wanted to show you in terms of some real simulations is one where this one is also leukemia-oriented. It's actually the same type of leukemia. Slightly different target. And there's a drug called dasatinib that's known to bind to this one, which is called Src kinase. It's an approved drug. And what Yibing Shan did, which had he asked me first, I would have probably said, well, Yibing, if you want to try it, of course, that's okay. But, I don't think this will ever work. I mean, it's probably a waste of Anton time. And I might have just said, look, why don't you do this instead? But he didn't ask. He just did it because it was a simple experiment. And what he did was he took dasatinib and put it in a simulated box of water. You can just see it peeking around here. It's actually not attached anywhere. And he just released it with no, fake non-physical forces, with nothing. Just let it swim around to see whether it could find the correct place where that drug actually binds on the Src kinase. And the interesting thing is that the place that it bound let me show it to you first, and then I'll say this. But it's going to move really quickly. It's going to be searching around, trying to find a home somewhere on this kinase. So this is yet a different kind of representation of a protein showing the surface. And it spends a fair amount of time initially there, but it can't quite figure it out. And eventually, it's locking into what turns out to be an extremely good fit. And the interesting part, well, there's several interesting things. The fact that it can find the correct binding site and bind as well compared to an experimentally known bound structure is interesting. But the thing that's really cool here is that the binding pocket, the thing it's fitting into on the protein, doesn't actually have the correct form until this ligand, this drug, induces that structural change. It's not a matter of the drug sort of moves by and finds the exact lock and key. It's also helping to create the correct fit. And they've co-evolved. And this drug didn't exist out there, but this is an area where signaling is going on, and some endogenous molecule, some molecule in the body itself, has presumably found a way to bind, but it causes that to happen. And that kind of induced fit is very interesting to me in particular. So let me show you one more of those, again, because it's fun. I think right now this other drug, which is involved in breast cancer, another approved drug, is hidden behind here, but you'll see the same thing. There we go. And this is a different representation that you've seen. Almost all of them, these are little tubes to show it. But you can see more, you don't see the shape filling part here, but this allows us to see what chemical groups are making contact here. This is sort of a greasy area right here. Just like, if you pour oil on the top of, a pot of water when you're making spaghetti and the oil droplets kind of come together, that's a hydrophobic effect, and that works with molecules too. Whereas some of the things over here are forming these special kinds of bonds called hydrogen bonds. Polar molecules line up so that they interact in a way that's actually fairly tricky. So we can, with this kind of representation, we can sort of see what's going on. But we're doing more studies of this sort now. I don't think it's the straightforward thing of, well, let's release a lot of candidate drug molecules into a box and see if they stick to the molecule of interest. That's not really practical. It takes long enough that you wouldn't do this with 1 million compounds. But in terms of studying the process of drug binding, the recognition of binding sites, it's quite interesting from that viewpoint. Maybe it'll lead to something good. And I'm just very grateful that Yibing doesn't do what he's supposed to be doing. Anyway, let me say a little bit at least. Ooh, I should have done this earlier. I'm supposed to stop talking in 5 minutes, I guess, and then we have questions? Or?
Around 5, but if you want to go a little over, it's fine. We have approximately half an hour for questions.
Okay, that's a long time. So the longer I talk, the less likely I'm going to be to get horribly grilled. I can fill up a lot of space when I'm filibustering. No, let me try and get through it and take some questions. So what makes Anton fast for this particular application? The first thing, although it's kind of a buzzword, is Codesign. This really is an example where when I came up with the basic idea for Anton, it was literally the same day when I finally was working out the end of the algorithm and the end of the architecture. They were mapped to each other, and if, at this point, now when we're designing Anton 3, actually, Anton 2 should be here in a year or something like that, I guess, a little more. But at every stage, when we're thinking about the architecture, we're thinking about the algorithms also. And Anton was designed to use a very different algorithmic approach, especially on the communication side, that was well suited to mapping things onto a silicon surface. If we had taken standard hardware and tried to come up with new algorithms, I mean, maybe we would have come up with some. There are a few things that we've done that are applicable to conventional clusters also, but we would never have gotten to this kind of speed. And conversely, if we had come up with, if we had designed hardware to take current algorithms, I forget if I said it, it's both directions, I forget which one I said first, but we couldn't take standard hardware and design algorithms or standard algorithms and design hardware for it and get anything like this kind of speed up. So a lot of it is mapping one to the other with a sense for how silicon can be used in a very different way if you've got a specialized problem. And I realized the way I just phrased it made it sound like this is some general principle, and I want to make sure nobody leaves with the impression that that we've discovered some magic way to design for any special purpose application. That's not the case. And sometimes I'm asked, well, what can you learn about designing special purpose machines for various problems? And I don't honestly know what you can learn from the Anton experience. In fact, one thing it's easy to say is there are many problems that are very important in scientific computation for which this kind of approach would definitely not work. And some examples are things where you can rigorously prove, in fact, that there's a bound on how much speed up you can get through parallelism, typically enforced by a communication problem. And so in those cases, it doesn't make sense to pursue the special purpose approach and try and get major speed ups. You'll speed up one part all the way, and then the thing that will hold it back will be something you can't do anything about. There are other cases where it's not so obvious this wouldn't work, but I honestly don't know what the range of applicability is. And even in areas that seem like they're the same thing, I visited several of the national labs and several other groups where people are actually doing MD simulation, and we'll talk afterward, and I'll say, oh, gee, you're doing MD simulation, maybe something like this can work. And what's almost invariably happened is when we went into the details, it actually wouldn't. You'll find out, well, their force field is a little different and takes a lot longer, and they're really not interested in the particles moving around because they have crystal structures, or they're studying metals, or they want this interface, or they study enormously large systems to see the kinds of things they're interested in, like cracks propagating through big bulk materials, and they're willing to simulate for very short periods of time, or some other things involving parameters or other things that look like details that, when you look at it, mean that you really can't apply this kind of technology there. So the bottom line is I'm not sure how much really applies. But the algorithm and the architecture are focused very heavily on executing particle-particle interactions very quickly, or particle-point interactions. It can either be two particles interacting together along the lines of the force field that I described, or particles interacting with a grid, a regular sort of lattice. And I won't describe why exactly, but there are a couple applications for that, for interacting a particle with distant atoms. I said that I was lying a little bit about interacting all pairs of atoms. If something is far enough away, then you can actually cheat a little bit and represent the very distant ones using some sort of a continuum approximation, either with some nice smooth Fourier transforms or representing it using some other kind of series expansion. There's the fast multipole method that you can use. You can use an iterative solution to the Poisson equation for distant things. There are a lot of different approaches that you could use. And in many of those, mapping things and interacting on a grid is an important thing to be able to do. So it's very fast for that. I'm going to skip over this except to say that, actually I'll get to that. Polarizability is something that I only alluded to briefly, but it's a defect in current force fields, known defect. Most of the ones that are out there make an assumption that's manifestly not true, which is that every atom bears a certain, typically partial charge on it, and independent, the model is that it depends on the immediate covalent neighbors, the immediate bonded neighbors, but not on the electrostatic environment. And that's just wrong. If you have an atom here and there's a big positive charge over here, it's going to sort of smush the electron cloud, I guess drag it toward it in this case, and cause charge to sort of be distributed in different ways. And it's amazing that you can get any decent results. I wouldn't have predicted you could get meaningful results without having that. But in fact, most people who do these simulations, including us, are using non-polarizable models. So it's designed, Anton is designed to accommodate variations in most existing force fields, to accommodate one particular model of polarizability and several other things. But it's still a very special purpose, really laboratory instrument more than what you could ordinarily call a supercomputer. So, okay, where's the underlying speedup from? It's really from a couple different things. One is that for the specialized calculations that Anton has to do for MD simulations, you have massive specialized parallelization, a bunch of really stupid, simple units that can only do one thing. It's basically like an n-body engine, something that can interact pairs of particles really, really fast, but that's all it can do. That's this part of the machine. And then there are places where you actually do need some flexibility and programmability, but you only want to do the things that you absolutely have to that way. And fortunately, this isn't because of any ingenuity on our part, although many members of the group are extremely ingenious, who come up with all sorts of things I would never have thought of. But we're lucky. We're very lucky that MD simulations actually have a property where the parts that you spend most of your time doing are very regular, mapped neatly onto silicon, and the parts that are less regular, or things that if you design special purpose hardware for, would be used a very low fraction of the total time, those things don't happen very much. Like the bonded interactions, there's math to do, but there aren't that many atoms that, a given atom isn't bonded closely to very many other atoms. The part that takes a long time is the interaction with all the other ones, which is very regular. The little idiosyncrasies of the local interactions, you can afford to do more flexible because it isn't the inner loop. It's not the thing that takes most of the time. So that's one thing. Oh yeah, the hardware, it's very dumb. Actually, I have a picture of that. And the only thing that's variable is some tables and a few parameters, but it's not, that part of it, the engine that does most of the work, is not programmable at all. There are other things that are sort of, there are other parts that are programmable, and they're pretty horizontal, and data flows in complicated ways, but it's very difficult to program, and it's not the source of the major power that we're using for the inner loop part. The other part is the communication, and the first thing to say is within the chip, the data is flowing in a highly regular pattern. You don't have things where it's going off and being stored in memory all the time and pulled out and then going random locations. A lot of things have a whole wave of data going through in a very regular pattern. So things tend to go only where they're needed at just the right time, and that makes it very easy to save on the control circuitry and to avoid memory bottlenecks and so forth. And then when it comes to outside memory. With a conventional microprocessor, a lot of the time is actually spent bringing data in from memory. And in fact, with a modern microprocessor, you go to all sorts of lengths to avoid having to do that very much, like keep stuff you've recently used on the chip and local faster storage. And even so, the memory, access to memory, is a big bottleneck. And basically, to a first approximation, Anton doesn't have to go to any external memory at all. Now, it's not quite true, but the inner loop part, all the data is residing on the chip. It all moves around here, and you don't have to go out. For very big systems on a small machine, you would sometimes, but it's designed to basically run with memory that's internal to the chip. And then on an inter-chip level also, the communication is handled in a very careful way, and that extends to two different aspects of communication, one of which is latency. And latency is just a measure of, even if I'm sending data from one processor to another, one chip to another, even if I have a tiny amount of data, suppose I want to send 1 bit of information from this chip to that chip, how long does that take? What's the sort of fixed overhead? That's the latency, how long it takes to get there. And then the other part is bandwidth. If I want to then continue pumping data down here to these other locations, and in fact have all of them send to all of the others or some pattern like that, how much data do I have to shove down all of those pipes? How fast can I do that? Both of those are critical, and we've given a lot of attention to minimizing the time required for those also. So I'm just going to quickly go through because I do want to hear some questions. Top level of the machine, this is how it's wired. It's basically a three-dimensional mesh, but with end-around connections. It's really a hypertorus. And so the data gets exchanged here, but if it falls off the right side, it goes to the left. And there are sort of two reasons. One is it's not exactly a reason, but this has been used. This interconnection topology has been used even for general purpose supercomputers. But in our particular application, with the method that we use in Anton 1, we are actually simulating not just one, conceptually, we're not just simulating one box full of water and protein. What we're actually simulating is an infinitely tiled array where this box is repeated like a crystalline structure going off to infinity. So all that really means is that if the system being simulated is distributed among these, say, 512 processors, if an atom runs off the right side of that box, it magically reappears on the left side of the whole box. So, it's what's called periodic boundary conditions. And that's nice. That has some good formal properties, which allows you to do some sorts of solutions to the distant problem. I won't go into it much more, but this is a convenient architecture for that reason and preserves the locality of space. I guess I should have said this directly. This is something where you divide space into a bunch of little cubes, and then the interconnection pattern mirrors the way those cubes are physically connected or proximate to each other in the machine. That models the space. I'm sure there's a more elegant way to have said that. Anyway, here's the stupid part that does lots of the work. This is what we call the PPIP, the pairwise point interaction pipeline. There are 32 of these on every chip. And I'm not going to go through exactly what they do, but a key thing to just notice here is that there's a lot of little blue boxes, and every one of these blue boxes is doing some arithmetic, something useful in the computation. And it's pretty much a linear pipeline. You're not seeing loops going around here. You don't freeze while waiting for things to happen. Just to give you a rough idea, the thing that they're more of than anything else is basically calculating a very low resolution distance between two atoms. All it's trying to do is filter out all the ones that are obviously too far away to be interacted in this part of this subsystem. And then the ones that are left, it's going to do a more accurate calculation, and then it'll do a bunch of other stuff. And there are table lookups and some stuff here that does cubic interpolations and things like that. All the take-home message is just lots of dumb things. And each one of these little units is very, very small. Multipliers, their width is proportional to the square of the bit width. So this one actually is, I believe now 8 bits. I designed it as, I think it was 6, but they wouldn't let me have something that wasn't a power of 2. So it's a little bigger, but that's a tiny little multiplier, and then all of these units are very tiny, and the data flow is very regular and so forth. So that's why you get a huge amount of arithmetic density. And I think I left out the flexible part just very quickly. Okay, what I just showed you, 32 of those, it's 28-stage pipeline. So that's where you get a lot of the speed. On Anton 1, that's about a gigahertz clock, but it's doing, at any given point in time, you're doing a lot of operations. I talked about the particle-to-particle and the polarizability, and I'm just going to run by these slides very quickly. Okay, what I'll say here is just for a given atom, I've sort of alluded to this, we're actually going to do all the pairwise quadratic number of calculations inside some box, and then we use other kinds of approximation, in our case based on a technique that was developed in the group, but is based on some of the things that many other people use, the use of Fourier transforms and a way of decomposing the interactions into 2 parts. What you're left with in this part is the n-body problem, classic n-body problem of physics, in a form where you often see it in computational science, which is a range-limited n-body problem, all pairs within a certain distance. And there's an algorithm I came up with that minimizes the total bandwidth of the communication rather than go through it and its heritage. Let me just show you graphically what it looks like. So here is one of those little cubes in space. That's the green box. And the traditional method for doing this was something where, okay, so here's a box, and let's say this atom that's in this box has to interact with all atoms within a certain distance of it. And that whole volume of where the atoms are that has to interact with some atom inside that box is shown in blue here. So it's pretty close to a hemisphere. That's the traditional method. You interact pairs of atoms inside the box where at least one of those 2 atoms live. And it sort of makes sense. You would want to do that. At least one of the atoms you don't have to move anywhere. But it turns out that's actually suboptimal. And the NT method, what it does is divides up the space such that if you had any pair of atoms, they always go to some box that, in general, neither one of them live in. They might by coincidence, but it turns out the most efficient way, both in absolute terms and asymptotically, is to have them meet on neutral territory, is the way I referred to that. And so you define one area, which I call the plate, and another one, which is the tower, and everything inside this place has to interact with everything inside this tower. And then as the number of processors or processor-like things, I want to explain exactly what I mean by that, goes up, the advantages go up. And in fact, if you had 64 processors, there really isn't one. This is the traditional approach. This is the NT approach. But if I go to 512 processors, and then 4K, and finally up to 32K processors, what happens is that this scales as the volume. So it's if the interaction distance is R, it's going to scale as R squared. And over here, what you're going to get is scaling with R to the 3/2, where with the number of processors, because this becomes an infinitesimally thin plate, and this becomes an infinitesimally thin rod going up there. Now, what really matters, somebody once told me, we don't live in asymptopia, and that's absolutely right. You have to look at the constant factors. Constant factors are very good for this application in Anton 1, and we'll be using something pretty much like it in Anton 2, but I've been, including much of last night, been spending a lot of my time thinking about Anton 3, which is another 5 or maybe even 6 years out in the future. and am busy trying to figure out ways to rip to shreds all the earlier assumptions. I think this general approach probably will not survive because now we're worried about a whole different set of constraints. The speed of light going through the wires between our chips is going to be one of the limiting factors in how fast we can accelerate MD simulations. Latency will become more important than bandwidth. We'll have tons of area on the chip, but the clock cycle speed won't speed up that much more. The world is changing gradually, but in distinct ways, and so we're trying to think out what's that next generation going to look like. And in particular, how are we going to change not the laws of physics, but the abstract models that we use? Are there some ways we can cheat? You know, it's becoming so difficult to import distant atoms to somewhere else. Does it really matter to get those effects very accurately, or can we do some things, as I suspect is the case, that are kind of even grosser approximations, but with functions that are wrong but well behaved, and where various things you can expect to be canceling out, and maybe spend more time on the local stuff? Don't know the answer to all of this, but it's going to change. The next generation, especially after this generation coming up, is going to look significantly different, or it won't represent a big improvement in speed again, and that's something we would like to see. Maybe not forever, but we'd really like to see a substantial increase in the length of time we can simulate. Are those lights saying please get off the stage? Or it looks better with them, but anyway, I'm going to pretty much close this, just say what each slide is rather than telling what's in it. Basically the way we get this, inside the chip, all of the particles in the tower are moving in this way. Yeah, that's kind of the other extreme, isn't it? And then the ones from the plate move here. With a linear amount of input, we have a potentially quadratic number of interactions. And then when it comes off of the chip, we have a reduction. They get combined. The forces all get combined together on the same particle, and then we're back to linear. So the amount of I/O, which has to be limited pretty strictly, is linear, but the amount of computation can be quadratic. That's just a sort of that's a very severe oversimplification. Last slide, which is about force fields again. I already mentioned this. We don't know just what the limits of current molecular mechanics force fields are. We do know now, and especially over the last year, I think we've provided pretty strong evidence that you can learn about a lot of things that weren't known before, understand a lot of processes through long simulations, even with today's generation of force fields. We don't know what the limits are, and one of the advantages is, even though some of the limits may be real limits, the nice thing is that we can simulate long enough now that hopefully we'll be able to discover what some of those limits are in a way that we couldn't have if we couldn't run such long simulations. Seems like a pretty, as I say it, it seems like kind of a lame excuse. We couldn't, if we run into limitations, well, at least we've found what the limitations are, but that's true. And there are actually some interesting things scientifically about that, because if we find out what the missing pieces are, where are the problems, biological problems. I'm not really concerned with the underlying physics. We know what those are, but does it really matter if we approximate this? Is there something else that has to be represented quantum mechanically? What matters in terms of biology is a non-obvious, at least to me, that is, the answer is non-obvious to that question. And this may be a naive way to think about it, but here's the way I think about it. A lot of it just has to do with how did proteins actually evolve or have to evolve? Is it the case that proteins are designed in a way that they have to be robust in the face of thermal noise, and most mutations are actually not ones that cause something horrible to happen. In the case of the silent mutations, maybe things are designed in a way that proteins are so robust that it's okay to calculate some of these things wrong in particular categories. Or we also know that proteins serve as switches. So they ought to have a careful free energy balance. In some cases, you want them to be sort of balanced like this. Something causes them to change, and it falls down this side of the hill. And then maybe you really want a subtle change to make it go down this side of the hill also. So maybe nature has conspired. Nature does a perfect example. They do a perfect job, or it does a perfect job of solving the Schrodinger equation. It's really, really good at that. But we can't do that. So maybe for it really is fine. It can be carefully balanced. If anything's wrong, it'll mess up our simulations, but nature doesn't care because it does it exactly. Maybe that's the case. And I don't know, I don't have a good enough intuition, if anybody does, to know which way that's going to pan out. But hopefully, by getting the answers to some of those questions, at least we'll learn something about biophysical theory. And if we're lucky, maybe we really will be able to simulate for long enough and through new force fields accurately enough that we can design a lot of drugs and save some lives. We'll see. We take some questions?